We are very pleased to have with us Leeana Peters from the Brusca Lab at University of Florida. The title of her talk is How Does Immunometabolism Influence the Partial Remission Period of Type 1 Diabetes? That can also be known as the honeymoon period. And a little about her bio. She completed her PhD in May 2023 with a focus on elucidating the role of T1D candidate genes such as PTPN22 and CD226 in disease progression, as well as utilizing bioinformatic approaches to identify novel cell subsets and biomarkers for type 1 diabetes. She joined the Brusca Lab as a first-year undergraduate student, received a Howard Hughes Medical Institute fellowship. That's awesome. For the 2015-2016 academic year to investigate the CD226 and the TIGIT axis in regulatory T cells and potential cellular therapy applications. I'm going to drop her recent paper in the chat. And we are excited to hear what she has to say. Drop some questions in the chat and we'll take them at the end. Thanks for joining us, Leeana.
Yeah, thank you so much, Monica, for having me. It's always a pleasure to talk with you guys. And like Monica said, I'm going to be talking today about the recent work that she just shared regarding examining tissue immune phenotypes and receptor repertoire in type 1 diabetes. So just going ahead and getting started. We all are really familiar with the fact that type 1 diabetes arises from a complex interplay of genetic and environmental factors. And kind of adding to this complexity is the fact that there are a lot of diverse cell types that have been implicated as well in the progression to type 1 diabetes. And these include, but are definitely not limited to, the islets themselves, as well as resident innate immune cells such as macrophages and monocytes, dendritic cells, adaptive immune cells, including T cells and B cells, as well as cells such as endothelial cells and resident stroma. And to date, I think there have been over 150 genomic regions which have been associated with risk for type 1 diabetes. And a large portion of these genes thus far have been associated with some role in immune cell function. So indeed, when localization of some of these single nucleotide polymorphisms or SNPs that confer risk to type 1 diabetes was examined across multiple tissues, including blood, pancreas, and thymus, and across a lot of different cell types, including cell lines and primary cells, it was found that a lot of these SNPs tended to localize within enhancer regions, specifically within lymphoid cells, including CD4 T cells, CD8 T cells, and B cells. So this implicates a role for genetics and type 1 diabetes risk SNPs in altering the regulation of gene expression in immune cells and thus contributing to pathogenesis in this way. So one of the key hallmark immune-related histopathological features of type 1 diabetes is HLA class 1 hyperexpression within the islets. And as you can see here, HLA class 1 tends to be preferentially upregulated within the residual insulin-containing islets in type 1 diabetes, as opposed to those that are insulin deficient and compared to controls. And this is thought to be mediated, at least in part, by STAT1 signaling. As you can see, this increase in STAT1 transcript within islets of T1D donors derived from the DIVID study as compared to controls. And one of the potential consequences of this elevated HLA class 1 expression within the islets is that this may result in increased visibility of the islets to autoreactive CD8 T cells. And indeed, CD8 T cells comprise a large component of islet infiltrate. And it was found in this very seminal study that CD8 T cells specific for various autoantigens such as IA2, IGRP, preproinsulin, and insulin can be found within the islets, and in fact can be found to co-localize with insulin-producing cells within the islets, thus implicating CD8 T cells as key players in islet destruction. Additionally, it's been shown that CD8 T cell signatures may be important to investigate as signatures of immunotherapeutic response. So in the teplizumab trial, it was shown that donors who responded to teplizumab or anti-CD3 possessed an increased percentage of these memory CD8 T cells that express multiple inhibitory receptors such as TIGIT and CD57, as well as the transcription factor EOMs, which is important for promoting terminal differentiation of these cells. And moreover, they found that this population of multiple inhibitory receptor expressing CD8 T cells, marked here as TIGIT positive, KLRG1 positive, were shown to be induced upon treatment, as you can see here with the bump after these treatment arrows. And the boost in this population was maintained across the study period in responders relative to people who did not respond as well to teplizumab. A very similar transcriptional network was found with treatment of alefacept as well. Here you can see this gene network that contains genes that encode these inhibitory receptors, namely LAG3, PD1, and TIGIT, as well, again, coinciding with this transcription factor EOMs after alefacept treatment. Moreover, with mass cytometry, it was shown that there were multiple clusters of related inhibitory receptor expressing populations defined by expression of PD1 or CD57 that were found to be increased in responders across the study relative to non-responders. So this indicates that there may be a tolerogenic function of this induction of partially exhausted CD8 T cells across multiple immune-targeted therapies in type 1 diabetes. However, it's really important to note that circulating and tissue immune phenotypes may not always correspond. So it's really necessary that we examine the cross-tissue relevance of immune phenotypes in type 1 diabetes. As one example of phenotypes that may converge across tissues, it was found that responses to autoantigen in the peripheral blood could be segregated into multiple groups, those defined by the production of immunoregulatory cytokine IL-10, as well as a reduced frequency of autoantibodies, and another grouping that was defined by the production of inflammatory cytokine interferon gamma in combination with multiple autoantibodies. And a related signature was also found within the pancreas itself, where these two histopathological patterns emerged, one defined by high CD20 infiltrate, so more B-cell infiltrate, and low CD20 or less B-cell infiltrate. And this was found to be correlated with CD8 T-cell infiltrate as well. So this sort of difference in hyperimmune versus pauci-immune response may be common between peripheral blood and tissue. Conversely, it's been shown that the frequency of autoreactive T cells may be pretty different between the peripheral blood and the target tissue, which is important for monitoring purposes. So here in this study, it was shown that the frequency of autoreactive, in particular ZNT8 positive T cells, did not differ across groups that were age matched. So between T1D adults and healthy adults, there were no difference, and the same for the age matched group of children. And this was the case across multiple epitope specificities, including more ZNT8, a specific PPI epitope, GAD, IA2, and IGRP, with the exception of one preproinsulin epitope. And it was also noted that this was not true in the pancreas. So within T1D pancreata, there's actually an enrichment of ZNT8 reactive CD8 T cells relative to controls in T2D, which was not shown in the pancreatic lymph node. The pancreatic lymph node, or the PLN, is thought to serve a really important role as a priming site in type 1 diabetes, where dendritic cells present islet-derived peptides to autoreactive T cells, which then results in propagation of the immune response. And the critical role of the PLN was shown through multiple studies, but firstly, the study that examined the role of excision of the spleen or the PLN on disease prevalence. So here, this is excision of the spleen, where we see no difference in disease prevalence at early or later time points, whereas early excision of the pancreatic lymph node resulted in a decrease in disease prevalence in the NOD mouse. Moreover, in the BDC 2.5 TCR transgenic model, it was shown that autoreactive T cells selectively are activated within the pancreatic lymph node and the islets, as shown by increased CD69 expression. And upon adoptive transfer into T cell deficient mice, we see selective proliferation of these autoreactive T cells within the pancreatic lymph node as compared to other lymph nodes in these mice, which indicates altogether that in the NOD mouse model of type 1 diabetes, the PLN environment is conducive to autoreactive T cell activation and function. More recent studies have investigated the role of the pancreatic lymph node in supporting continual islet destruction. So this publication used transfer of naive IGRP-specific T cells into immunodeficient hosts, which then resulted in these two populations, a PLN-derived population expressing TCF1 and a pancreas-derived population of IGRP-specific T cells with low expression of TCF1. And these populations differed in that the TCF1 high cells from the PLN expressed increased genes that had to do with self-renewal and downregulated genes that were involved in activation and exhaustion, as well as effector molecules, indicative of their role as more of a progenitor population. And indeed, when these cells were adoptively transferred again into immunodeficient hosts, what they saw is that low numbers of this precursor IGRP-specific population could transfer disease, whereas much higher frequencies of the TCF low, more terminally differentiated cells could not, indicating that this population that is maintained within the PLN is particularly important for sustained autoimmunity. Human organ donor studies have also shown that the PLN in health may contribute to Treg function and support Treg function, and this may be defective in type 1 diabetes. Here, as an example, it was shown that Tregs derived from the PLN of T1D donors were less efficient suppressors of polyclonal T responder cell proliferation compared to those derived from control donors. And this was also true in that they were inefficient suppressors of proinsulin specific responder T cell proliferation as well. In examining phenotype within the PLN, it was shown that there may be some differences in inflammatory versus immunoregulatory profiles, where within the T1D PLN, we see more of a shift toward a Th17 inflammatory phenotype, and in controls, we have more immunoregulation in the form of Tregs. Moreover, it was also shown that Tregs from T1D PLN express an increased amount of a particular microRNA 125A5P, which was thought to target CCR2. And in fact, Tregs from the T1D PLN expressed reduced CCR2 expression relative to peripheral blood, and this was shown to correlate inversely with expression of this microRNA. So this altogether indicates that in human PLN, the environment is also not conducive to Treg function and may not support Treg migratory function from the PLN to the islets. So as in the field, we've had some great success in translating immune therapies that provide disease delay in the clinic, but it's worth noting that thus far, cell-based therapies, namely Treg adoptive cell therapies, have not been effective in type 1. And this points to an existing knowledge gap in terms of what subsets Tregs may need to suppress in tissue, as well as any milieu that may promote Treg destabilization or impede their function. Regarding overlap of repertoire within tissue, it's known that there are some cell type specific differences across tissues. Previously in our laboratory, we've conducted TCR beta sequencing as well as IgH sequencing of sorted T cell and B cell subsets derived from different lymphoid tissues from the Network for Pancreatic Organ Donors program. And what we see is that largely the repertoires of all of these cell types are pretty tissue specific, but CD8 T cells display the largest degree of overlap across tissues. And this was found to be true when comparing across lymphoid tissues, as well as comparing cells derived from the peripheral blood and cells derived from the islets, where you can see again that CD8 T cells tend to have the highest degree of overlap across all of these tissues as compared to T conventional CD4 T cells and CD4 Tregs. And this was derived from one donor. So given all of this data, we had some key questions that we still wanted to address. And these include, how do immune phenotypes in the PLN differ in type 1? What might be some of the molecular drivers behind altered phenotypes in type 1? And then how does the adaptive immune repertoire of T cell subsets and the associated phenotypes of these cells change between the pancreatic lymph node and the pancreas? So to start off, I'm going to talk about our mass cytometry data. So we conducted mass cytometry analysis of pancreatic lymph node from 10 T1D and 12 control organ donors. And we used a 35 marker immune panel to do this. This was done in multiple batches. So after performing normalization and batch correction, we performed unsupervised clustering of live intact CD45 positive cells from the PLN. And as you can see here, we broadly obtained clusters corresponding to CD4 T cells, CD8 T cells, B cells, myeloid and NK cells in this data. And we wanted to examine if there were any markers that differed across these clusters between T1D and control donors. So we performed differential expression analysis per cluster. And what we found is enhanced expression of the chemokine receptor CXCR3, which was upregulated in T1D donors compared to control donors in this particular cluster 4, which if you look at the dot plot above, you can see is a CD8 T cell cluster expressing the naive markers CD45RA and CCR7, CD45RA and CCR7, as well as the co-stimulatory markers CD27 and CD28 and CD127 as well, kind of implying a naive-like phenotype. So next we wanted to examine CD8 T cell phenotype at higher resolution. So we took these bulk CD8 T cells from the CyTOF data and clustered them again alone. And this yielded 8 clusters, which spanned from naive clusters to activated clusters to more terminally differentiated effector and exhausted clusters. And here we wanted to see if there was any difference in abundance in any of these clusters in T1D donors compared to control. And when we performed differential abundance analysis, what we ended up seeing is that there was an increased proportion of this cluster 0 population in T1D donors as compared to controls in PLN. And again, if you look at the dot plot, cluster 0 is indicative of a naive-like population marked with CD45RA, CCR7, co-stimulatory molecules CD27 and 28, IL-7 receptor, CD127, as well as low amounts of CXCR3 expression. And we were able to confirm this increase in an independent cross-sectional flow cytometry cohort of PLN donors, again from the NPOD program, where you can see an increased percentage of CXCR3 positive naive CD8s within the PLN in T1D donors. So this finding was of particular interest to us because we recently conducted a study where we had cross-sectional flow cytometry data from a very large cohort of peripheral blood, and we examined some of the phenotypes which most highly differentiate T1D donors. And you can see one, if not the most significant finding was CXCR3 low CD8 naive T cells within the peripheral blood. So this indicates that this phenotype may be important in both the peripheral blood and the target tissue. So CXCR3 is the receptor for IP10 or CXCL10, and previously this has been shown to be elevated within the serum of newly diagnosed T1D subjects as well as high-risk subjects. And this was shown to correlate with the amount of interferon gamma in the serum as well, with it being elevated in high risk and newly diagnosed type 1 diabetes donors, which is in line with both of these IP10 and CXCR3 being interferon gamma inducible. Notably, within the islets, it was also shown that CXCL10 is elevated as well as CXCR3 in the islets of T1D donors compared to controls, indicative of ongoing inflammation and continual recruitment of immune cells to the islets. Notably, CXCR3 expression on naive-like subsets is indicative of a TSCM-like profile. And these cells are characterized by the ability to self-renew as well as to generate more terminal subsets such as central memory, effector memory, and exhausted populations. So having identified this increase via flow and cytoph in this naive-like, potential stem-like population in the PLN, we next wanted to examine the molecular drivers of this phenotype. So we performed single-cell RNA sequencing on the pancreatic lymph node of 9 T1D donors and 7 control donors. We used the 5' kit from 10x Genomics in order to obtain both transcriptomic and TCR sequence information from these donors. And again, just broadly showing the phenotypes that we obtained within the PLN, we have clusters of T cells, B cells, and innate immune cells such as macrophages, ILCs, and DCs. Again, we're interested in examining T cell phenotype at higher resolution, so we subset these T cells and reclustered them in order to obtain higher resolution profiles. And you can see we have a diversity of T cell subsets here spanning from naive to different helper subsets, effector subsets, and exhausted subsets as well. So we focused in on CD8 T cells, and we wanted to examine differences in gene expression between T1D and control donors across the trajectory of CD8 T cell differentiation. So we looked at naive CD8 T cells, effector T cells, and exhausted T cells. And the differential expression analysis, starting from the naive CD8 T cell cluster, revealed an enhancement in inflammatory cytokine signaling, including interferon alpha and interferon gamma, as well as lymphotoxin beta, SOX3, IL-32, and IL-27RA. We also noted an upregulation of genes which are involved in restraining terminal differentiation, including BCL3, BCL6, and BATF. In addition to this, looking at genes which are downregulated, we noted a downregulation of genes that are involved in dampening cytokine response. So those would be TNF-AIP3 and SOX1. And we also noted a downregulation in genes that are associated with terminal differentiation or exhaustion, such as DUSP2, NR4A2, and TSC22D3. Within effector CD8 T cells, we noted an increase in effector genes, notably NKG6, NKG7, and HOPX, as well as an increase in genes associated with migration, VIM, and ITGA1. We also noted an increase in the expression of BCL2, which is really important for long-term maintenance of memory subsets. And looking at downregulated genes, again, similar to the naive cluster, we see downregulation of genes that are involved in promotion or association with exhaustion phenotypes, CXCR4 and NR4A1. So looking at, lastly, the exhausted cluster, we noted upregulation of genes involved in chemotaxis, such as XCL2, calcium signaling here, as well as, again, inflammatory cytokine signaling, and genes, again, associated with exhaustion. Notably, HLA class 2 was downregulated, as well as NR4A2 and DUSP2 were downregulated in exhausted CD8 T cells in type 1 diabetes. We performed reactome pathway analysis using these differentially expressed genes, and common to naive and CD8 T effector cells, we saw an enrichment in interleukin signaling, as expected. And then within exhausted CD8 T cells, we saw interleukin signaling as well as interferon signaling was enriched in type 1, and then also interleukin 21 signaling was enriched within exhausted T cells in type 1 diabetes. So lastly, we wanted to infer what the milieu might be contributing to these differentially expressed genes. So we used a tool called immune response enrichment analysis, or IREA, and this basically uses a database of genes which are known to be upregulated or downregulated upon specific cytokine treatment, so that you can infer what the milieu might be contributing to your profiles. And here what we found is that across all of the clusters here, IL-15 was the most significant, if not the second most significant potentially, contributor toward these phenotypes. And importantly, IL-15 is essential to maintenance of stem-like phenotypes and prevention of exhaustion. IL-15 has also previously been associated with type 1 diabetes pathogenesis, as in the NOD mouse, IL-15 increases along with disease progression. Moreover, in the NOD mouse, again, if you block IL-15 signaling, you get a delay in disease. And most importantly, within the islets of T1D donors, it was shown that IL-15 was increased as compared to control and autoantibody donors, and the receptors for IL-15 were also increased within the islets of T1D donors, implying that this pathway may be really important across mouse and human for pathogenesis. Related again to IL-15, it's been shown multiple times now that autoreactive circulating T cells may be enriched for a TSCM phenotype across multiple specificities. And epigenetically, it was found that autoreactive T cells possess this hybrid-like profile where they maintain accessibility at loci which are important for stem-like cells, notably TCF7, and they also have accessibility at some effector loci, such as interferon gamma and perforin. So I've discussed a lot of our gene expression analysis up to this point, but we were also really interested in performing repertoire analysis using the TCR data that we collected from these single cells in PLN. I just wanted to talk a little bit about basic repertoire analysis. So typically, this involves looking at differences in gene usage, as well as differences in diversity or clonality. But there have been some really important advancements in these analyses, which enable things like clustering of TCR sequences, as well as co-clustering of TCR and transcriptomic data. And lastly, and more complexly, machine learning analyses, which enable potential prediction of disease or antigen specificity. So for our analyses, we wanted to utilize a tool called conga, which examines the association between gene expression and TCR features. So essentially, it's clustering data based on gene expression and TCR, and then it's determining basically where these graphs overlap in terms of common gene expression and TCR features. So you can examine how gene expression and TCR may be interrelated. So we performed this analysis using our gene expression and TCR data from the PLN. And I want to explain a little bit about the map here. So this is the same UMAP that I've shown previously and overlaid it here so that you can see. This time it's colored by conga score, and really this is just indicating the degree of significance of each of these clones in terms of their overlap between the gene expression and TCR graphs, where the more yellow the color, the more significant the clone, indicating that the overlap is more likely to occur than chance. So here you can see that what we call our conga hits can be found primarily in CD8 T cell clusters, but there are a few that are located, although less significant, within CD4 T cell clusters. And here I've also included the conga cluster overlay on our existing gene expression UMAP, as well as our TCR clustering data, so that you can see that we obtain a large number of clusters of TCRs that are not just one clonotype. So these are comprised of a good number of sequences per cluster. So we wanted to examine if there were any differences in the localization of these conga hits per cluster across control or T1D donors. And what we found is that there was a cluster here, cluster 10 in gene expression and cluster 3 in the TCR clusters that was enriched within T1D. And I'll explain the identity of that cluster below. And bear with me because this map is a lot, but I'll go through it pretty slowly. So here on the far left are semicircles indicating the gene expression cluster identity and the TCR cluster identity. So our cluster of interest is here, cluster 10 for gene expression, cluster 3 for TCR. These bar graphs here indicate the composition of the data set as a whole or each individual cluster per status. On the far right is the composition of the whole cluster or the whole data set, where you can see it's 50-50 between control and T1D donors, indicating that there's no bias from the beginning in terms of the amount of clones derived from control or T1D donors. But there are differences when we look at the composition of clones derived from control or T1D across these clusters. So the one that we pick out here as being significant, cluster 10 in gene expression, cluster 3 in TCR, you can see that by these top 9 differentially expressed genes, it's a CD8 T cell cluster. It may be a little bit restricted in its TCR alpha gene usage here, and maintains a pretty diverse CDR3 motif in terms of the TCR alpha region, and it's very diverse still in terms of the TCR beta motifs. Notably, this cluster is pretty large. It's comprised of about 100 cells, so there's not an overwhelming bias. And then if we look at the marker genes on the far right, you can see that it's a little bit hard to see, but this cluster still expresses CCR7 and cell, as well as IL-7 receptor. So again, supporting the fact that we have this enrichment in diverse CD8 T cells within the PLN in type 1 diabetes. So given the existing mouse data showing that the PLN is important for sustaining this autoimmune population that self-renews and gives rise to more terminal effectors in the pancreas, and our existing data thus far showing that this phenotype may be relevant to human tissue, we next wanted to examine the interplay of these phenotypes across tissues, notably the PLN and the pancreas. So to do this, we collected pancreatic slices and performed single-cell RNA sequencing, again, using the 5' protocol in order to obtain transcript and TCR for these. And this was done on 2 donors for whom we had paired PLN data. And they do differ a little bit in disease duration. So donor 6551 is a pretty notable donor with low disease duration, 0.58 years. Donor 6536 is higher duration of 4 years. And you can see just with these images from the pan-tail that there's differences in insulitis between these donors, where there's more severe insulitis in the shorter duration 6551 donor. So what we did was we integrated the PLN data with the pancreatic slice data. And here you can see the clusters that we obtained after integrating PLN and pancreatic slices. And they range across different T-cell phenotypes, as well as inflammatory macrophages, B-cell cluster, as well as some pancreas tissue from the slices. So we're interested in examining the differences in distribution of expanded clones across these clusters. And what we found is that the majority of clones that were expanded, being all of the colors other than this black bar, which represents singlets, were found within CD8 T cell clusters, notably the CD8 T effector and the CD8 cytotoxic T lymphocyte cluster. And given the sparsity of single-cell data, as well as the heterogeneity of these tissues, expectedly, we don't have a great degree of overlap in repertoire across tissues, as shown by the low Morishita-Horn index here. But despite all of the technical challenges, we were able to obtain some clones which were shared between the pancreatic lymph node and the pancreas. And these included primarily CD8 T cells, and there was sharing between clusters, cluster 0 and cluster 2, and there was some sharing across tissues, but again, the majority of the sharing across tissues was within CD8 T cell clones. We do detect, I believe, one CD4 T cell clone, which was expanded and present in both PLN and slice in donor 6536. But looking at the differences in phenotype across tissues, what we found expectedly is that within the pancreatic lymph node, there tended to be more naive cells, and the clusters which were shared across tissues tended to be more differentiated effector cells. So clusters 0 and 2, you can see, are both present in the PLN and the slice, which are the CD8 T cell clusters. Additionally, overrepresented in the slice are some of the inflammatory innate cells, such as IL-1 beta macrophages. So again, we're interested in looking at CD8 T cell phenotype and how it differs across tissues. So we performed differential gene expression analysis using total CD8 T cells and looked at the difference in expression between PLN and pancreatic slices. And notably, a few genes came up which promote stemness and naive-like phenotypes, and these were LEF1 and CEL, and those were upregulated in the pancreatic lymph node relative to CD8 T cells from the pancreatic slices. But this could be driven just by changes in the proportion of naive and memory cells across these tissues, so we next wanted to conduct differential gene expression analysis on clusters which were shared across tissues, these being the CD8 T effector cluster, or cluster 0, as well as the CD8 CTL cluster, or cluster 2. So when we examine these differentially expressed genes, what we find is that within the CD8 T cells from pancreatic slice in this cluster, we see an upregulation of effector-like genes such as interferon gamma and granzymes. And we also see upregulation of exhaustion-related gene DUSP4. We also see downregulation of common gamma chain cytokine genes such as IL-7 receptor and IL-2RG. And this was, again, very similar with the CD8 CTL cluster, where we see upregulation of effector genes in slice and downregulation of common gamma chain cytokine genes in this cluster in slice. So lastly, we wanted to use this data to perform gene set enrichment analysis. And what we found expectedly is that within the cluster 0 on top here, within the pancreatic slices, we see an enrichment of inflammatory cytokine signaling, including IL-6 and TNF-alpha signaling. Whereas in this cluster within the PLN, we see an upregulation of pathways involved in promoting stem-like phenotypes, such as the Wnt to beta-catenin signaling pathway. And again, this was similar for cluster 2, the CD8-CTL pathway, in which we again see within slices, they tend to be more effector-like in inflammatory signaling, including IL-6 here as well, and TNF-alpha. So altogether, this indicates that the shared clusters across PLN and pancreas, those CD8 T cells from slices tend to be more effector-like, whereas the PLN maintains that stem-like profile. So just to conclude overall, we found that CXCR3 expressing naive CD8 T cells were enriched in the PLN of T1D donors. We found that these CD8 T cells in T1D PLN express reduced terminal differentiation-related genes, and they maintain effector gene expression throughout the trajectory of CD8 T cell differentiation. We found through integrative TCR and gene expression clustering that there was an enrichment in naive-like diverse CD8 T cells in T1D. And we think that IL-15 may be an important signal in promoting these phenotypes. We also saw through paired PLN and pancreas transcriptomic profiling that shared populations tend to express more effector phenotypes in the pancreas, likely due to a more inflammatory local cytokine milieu. And we also think that this data may have important considerations for immune-targeted therapies where, again, exhaustion has been considered tolerogenic. So moving forward, we're interested in how T1D risk loci impact these phenotypes in circulation and in tissue. We're also interested in how T-cell receptor affinity may play into these stem-like and exhaustion-like phenotypes, especially in T1D, where we're in the setting of chronic autoantigen exposure. And then, pertinent to the immune therapies point, what do these populations look like after targeting with therapies that may be relevant to some of these pathways, like JAK-STAT inhibition, or CXCR3 antagonists, or tibulizumab. So I just want to end by thanking everybody in our lab for being extremely helpful and supportive. I want to thank our funding sources and, most importantly, the donors without whom none of this research would be possible. So thank you, and with that, I'm happy to take any questions.
Thank you, Leeana. Congratulations. That's like an incredibly detailed body of work. So just wow. We do have some questions. Fatuma Samasa, she's asking, can CXCR3 by itself define stem-like autoreactive CD8 T cells? What about TCF1 or LEF1? Have you analyzed all these markers together? What is the link between CXCR3 and IL-15? Could you please comment? And Fatumata, if you want to unmute yourself to talk to her directly, that's great too.
Yeah, so I can answer some of that stuff. So we're now in the process of trying to validate exactly what you suggested. Is the panels that we had did not have TCF1 and LEF1 included. So that's something that we're trying to validate now to just make sure that it's really a stem-like population. It's definitely important. The link between IL-15 and CXCR3, so there's kind of a circuit that is sort of a positive feedback loop where I think interferon gamma can stimulate IL-15 and IL-15 can stimulate interferon gamma, and then gamma can upregulate CXCR3 as well.
Thank you.
Mhm.
Okay, great. Have to step out. Oh, but excellent work from Alok Jaglakar. Congrats, Leanna. Tinan Hassan Laswani, do you see a difference between stem like CD8 T cells and stem cells, memory CD8 cells? When you talked about how IL-15 maintains the stemness.
Yeah, so maybe he's speaking about sort of the way that I've defined them thus far as stem-like, just because,
Yeah.
like I said before, we don't have all of the markers to define them. If we do see a difference in LEF1 and TCF1, then I'll make that distinction that they are TSCM. I will say that other people have seen similar results to this, and I think there have been differences in things like TCF1 and TOX, which promotes exhaustion. So we're hopeful that these are truly TSCM, but certainly there's probably a lot of heterogeneity within the naive CXCR3 pool.
I had a question for you regarding targeting the PLN. There's no real sort of specific and temporal way to target the PLM. So for instance, if you wanted to do IL-15 blockade, if you wanted to do even CXCR3 blockade. I mean, one strategy, I guess, is a nanoparticle, right? Some kind of nanoparticle with surface modified with sort of like some kind of PLN homing ligand attached or associated. Have you guys considered that kind of experimentation down the line and thinking just sort of forward?
Yeah, that's a really good question. We definitely have some people here who could help out with that in the BME department. Getting a marker that is selectively expressed in the PLN as opposed to other lymphoid organs may be a little bit difficult, but
Like a
I know,
CCR7 or something, or anti-selective, I don't know.
yeah, they'll, I
They'll
think,
go other lymph nodes too, right?
yeah, but I know there are some other people working on some interesting things with looking at different metalloproteinases that are selectively expressed. So it's possible to do something like that. But yeah, I do wonder, you're right about the temporal thing as well. Like if we inhibit at a certain part before seroconversion, or do we
Yeah.
need an autoantibody to be present? So there's a lot of considerations about when you would need to target that stem-like population for sure.
Mm. Curiouser and curiouser.
Yeah.
Okay, Leanna, this is so exciting. University of Florida remains one of our favorite places in terms of all the things you guys are doing, especially the Brusco lab just has such a deep understanding of the immune side of the equation. And it's really exciting to see all the work that's flowing from the laboratory there. And congratulations again. We look forward to your next paper because it
Thank
sounds
you.
like you have tons and tons of data.
We have a
Fantastic.
lot of ideas, yeah.
Great. Fantastic. All right. Well, have a great rest of your day, and we look forward to another Briscoe Lab talk later this week.
Thank
Okay.
you.
Bye.